Well, good morning, everyone. Thank you all so much for joining us today. We've had over 180 people sign up for this webcast, so it should be great today. My name is Josh Forsyth. I'm the Vice President of Sales and Marketing here at Golden Helix. For the past 12 years, Golden Helix has been accelerating life science research by providing analytic software and services for a range of genetic applications, including genome-wide association studies, copy number analysis, next-gen sequencing, predictive modeling for diagnostics, and more. I'm grateful today to be joined by one of our customers, Dr. Matthew McClure of the U.S. Department of Agriculture's Agricultural Research Service. Matt will be presenting his latest research today on identifying causal mutations in agrigenomic species, more specifically in Weaver syndrome for cattle. A little more background on Matt and ARS before we start. ARS is the principal in-house research agency of the USDA with a workforce of over 6,500 employees, 2,200 of which are scientists working on nearly 850 research projects at over 100 locations across the U.S. and 4 overseas laboratories. Dr. McClure is a member of the Bovine Functional Genomics Laboratory of the USDA-ARS, whose mission is to improve the genetic and productive efficiency of cattle through research on gene expression and marker-assisted genetic selection. A little bit more on Matt. Matt received his PhD in genetics from the University of Missouri Columbia. Most of his research has a producer application side to it with a focus on identifying ways to apply cutting-edge genomics in a manner that benefits agricultural producers and ultimately consumers. His research has ranged from showing that DNA from FTA cards is high enough quality for SNP chips to identifying new haplotypes to predict meat tenderness to today's talk of trying to identify the causative mutation for Weaver syndrome so producers have an improved genetic test they can use to select breeding stock. Before I hand the presentation over to Matt, I'd like to point out the question and answer pane on your right. If you have any questions during the presentation, please feel free to enter them there. At the end of the presentation, we will answer as many questions as we can in the time allotted. If you're experiencing technical difficulty, please answer or submit your questions via the Q&A pane as well. One question that always comes up is, will we be sending out the slides and link to the recording? The answer is yes, barring any technical problems with the recording. We usually get that out within a day or two. With that, I'll hand the presentation over to Matt.
All right, thank you very much, Josh. So as he mentioned, I am a research geneticist, specifically a postdoc with the United States Department of Agricultural Research Service. And today I'll be talking to you about how with all the advancements in genetics, how we've been able to speed up the hunt for both causal mutations in cattle and how that's going to be applied to the species. So the ARS is the chief scientific research agency of the Department of Agriculture. We have over 800 research projects, over 8,000 employees, and we work all across the United States in 100 different research locations. We have 4 overseas locations. For the ARS, pretty much all of our work is farm-to-table research. So all of it has applications both to producers and ultimately to consumers. I am located at the Beltsville, Maryland, at the Henry A. Wallace Beltsville Agricultural Research Center. The BARC, as it's called, it is the largest and most diversified agricultural research center in the world. We have over 6,000 acres and just under 2,000 employees here. Of our employees, over 300 of them are permanent PhD scientists. We have over 100 postdocs and visiting scientists from around the world. I am part of the Bovine Functional Genomics Lab here in Building 200. We have over 12 PhD scientists, and our research covers everything from mapping to bioinformatics in cattle, to mastitis, genomic selection, to parasite resistance. Pretty much any way that we can use genomics to help the dairy and the beef producers, that's what we're interested in. Just to give people that are not familiar with the USDA some background, here's some of the highlights of some of our research. So about 150 years ago, the USDA was created, and in the 1900s, the first live vaccine for cholera for hogs was developed here. Starting in the 1910s, the ARS started to develop studies to improve dairy cattle. We also domesticated the wild blueberry. So if anyone has blueberry in the cereal in the morning, we were a part of that. In the 1920s, ARS scientists helped found population genetics and animal breeding. In the 1930s, the Line 1 Hereford program was started. So the animal L1 dominant that was used for sequencing of the cattle genome, her pedigree dates back to this Line 1 Hereford program. In the 60s, our scientists hoped to discover the molecular structure of tRNA. In the 90s, we helped develop genetic maps of blueberry, cattle, swine, chickens. And in the 2000s, we helped to develop the dairy cattle genomic selection. And we've also been sequencing lots of genomes. We've helped sequencing the cattle, chicken, swine, soybeans, turkey. The list keeps growing. So today I'm going to talk to you about how the technology advancements that have been used in genomics, what they are, and then how that can be applied to cattle. So some of the advancements in genomics are computing, genetic maps, sequencing, advances in genotyping, and eventually in analysis. So in computing, almost 100 years ago, the way we did computing and genomic research was by hand. Here at the USDA, we actually had hundreds of people employed where by hand they calculated the how good a dairy bull was. Luckily today we have moved on to using blades and servers for that research. For genetic mapping, especially in cattle, we have been able to move from having the linkage maps and marker maps of the 90s onto having the radiation hybrid maps, and then eventually we have the sequence of the animal. Genotyping and sequencing. There's still research done today where we use RFPLs for genotyping. We also have research where we use microsatellites. This takes a lot of time. For my PhD study, we ran hundreds of microsatellites over thousands of animals, and it took quite a few years. Today, we can use the high-density SNP chips. With these, there are scientists around the world that can genotype thousands of animals in a month for thousands of genotypes. We also are likely to have advancement in sequencing. It used to be that you could have your entire PhD project based upon sequencing one gene using the radiation gels. With the advancement of Sanger sequencing, and now with the next generation technology, you can sequence an individual's entire genome in a week. And finally, analysis. With all these other advancements in computing and in genotyping and sequencing, we're really lucky that there have been advancements in the analysis, from the algorithms to the models to the software. This is where Golden Helix comes into play for us. And all this analysis, what it is doing, of course, is just taking all this data and then filtering it into a usable result. So a classical example is identifying a QTL for a trait, putting that QTL on the map, eventually sequencing, and then finding the causal mutation that's either causing a disease or influencing a trait, anything from cattle to corn to humans. So how has all this been used in cattle? What has been some of the advancements in cattle genetics? All these advancements have helped quantitative genetics, phylogenetics, genomic selection, and identification of causal mutations in cattle. If we look at the number of cattle QTLs that have been published over the years, we can see a large spike in the past couple of years. If we overlay some of this technology, we can see that a lot of this increase has come about because of the advance in our genotyping and sequence maps. We've also been able to use the advancements in genotyping for phylogenetics. Jared Decker at the University of Missouri used the Bovine 50K panel, and he was able to improve the phylogenetics of the pecorine family. And not only was he able to improve the phylogenetics across species, he was also able to improve the phylogenetics across breeds. So he actually was able to show how the different breeds are related genetically. And what's nice with the phylogenetics is this means that you can take an animal and genotype it, and if you believe that it's an mixed breed, you can actually pull out what cattle breeds were in its ancestry. And finally, genetic selection. So for the genetic selection in cattle, the idea is how can we find the best cattle, either the best dairy bull that will have the best milk qualities, or the best beef animal that will produce the best steaks for us. Traditionally, the way that this was done for the past 30, 40 years has been using progeny testing. So for the dairy industry, you want to know how well his daughters will produce milk. So this means that when a bull calf was born, you had to wait 5 years. You had to feed it this entire time and wait till he was sexually mature so that you could breed him to a random group of females. And then you had to wait for those females to give birth. You had to select the females from that, wait 2 years until they were sexually mature, breed them, and then finally start collecting milk phenotypes. So this took 5 years and about $50,000 per animal. Because of genetic testing with genomic selection, with advances that have been performed by our lab and some other labs in the country, you can now genotype an animal. In about a month and a half, you can have estimates as to how well his progeny will do. And it will cost you less than $300. The advancement of this genetic testing is that the moment an animal is born, you can take an ear punch and you can send it out for testing, and you automatically get information as to how well his offspring will do. And the best way to explain this is that the information you get back, it's like you're gaining information on his future daughters. So at birth, for net merit, the amount of information you gain, it's like he automatically had 12 daughters that were producing milk. So this genomic selection using the SNP chips was started about 3 years ago in the dairy industry. Pretty much today, any top dairy bull is genetically tested for the SNPs and has genomic selection on it. And the benefits is instead of paying $50,000, today you can use the 50K chip, the high density SNP chip, or the new 6K SNP chip. And you can spend anywhere from $50 on up to $250. And one of the benefits is not only are you getting that information of the daughter equivalents, but using this genetic test, you're also increasing the reliability or the accuracy of the test. So if an animal is born, you take the average of a trait of his parents, the reliability is 42%. If you take that animal and you take the parent average and you do a 50K SNP chip on it, the reliability for any trait goes up to 72%. So automatically, not only are you increasing the number of daughter equivalents data, you're increasing the reliability for any of the phenotypes. And finally, all this genetic advancements has also helped us find the causal mutation for diseases. So if we look at 1985, the first causal mutation for genetic disease in cattle, inherited gold error, was found. Took 5 years later for the next one, and pretty much another 6 years before it really took off. Once we had the direct assembly and then started having all the SNP chips, the rate of identification of these causal mutations has really taken off. And this has really helped in my research today on the Weaver cattle. So with that, I'll get into my current research on looking for the causal mutation of Weaver syndrome in brown Swiss cattle. First, I'll start off, I'll give everybody a little bit of background on the disease, talk about the study design, and what our current results are. So Weaver syndrome, or progressive degenerative myeloencephalopathy, it is an inherited recessive disorder in the brown Swiss breed. There is destruction of the spinal cord and changes in the brain. This was first identified in the U.S. cattle herd of brown Swiss in the 1920s. So the clinical diagnosis of brown Swiss cattle, basically at 6 to 18 months of age, they start having onset of they have weakness in their back legs. It's been described as though they have trouble finding their legs. As it progresses over the next year to 2 years, they have more and more trouble walking, and eventually they can't get up. And so eventually, about 2 to 3 years of age, they actually die because they can't eat enough food. And one of the unique things is that the rest of their motor skills and sensory reflexes are fine. The symptoms from this disease, there's actually a degeneration of the nerve passages in the spinal cord in the brain. And from best that we can tell, this degeneration prevents nerve impulses from the brain to the back leg muscles. The nerve degeneration is actually comparable to Lou Gehrig's disease. So there's been hopes that eventually the advancements in the Lou Gehrig's disease research would help Weaver syndrome. This disease has actually been studied for quite a while. Michel Georges actually mapped it back in 1993. So he actually mapped it to chromosome 4 in cattle, and he actually was able to develop a diagnostic test. He found out that the satellite marker TGL116, it was in equilibrium with the causal mutation, and he estimated at that time in '93 that the microsatellite marker was 3 centimeters away from the causal mutation. About 5 years later, the locus was redefined, and it's actually down to a 10 megabase region now between microsatellite marker BMS 2646 and MAF50. And the genetic test that has been developed, animals are actually called at a 90% confidence level. So they're called either a carrier or a non-carrier. We actually still identify animals if they have affected offspring as a carrier as well. Now the issue with this is that it's not perfect, but it works well. So this is an example of you don't have to have the gene. You don't have to have the causal mutation to have an impact. This microsatellite marker test really helped to clean up the incidence of Weaver syndrome in the brown Swiss breed. So even though it wasn't perfect, it was extremely helpful and used in the industry. But there have been some historical problems with Weaver. First off, it's an imperfect commercial test because, as best as they could tell, it was a microsatellite marker test. It was a couple centimorgans away from the causal mutation. You will have recombinations, and because of the recombination event, you will have false positive and false negatives. One of the other problems is historically few, if any, females are tested. The males were tested, especially the males that would be used for artificial insemination. But because very few females were tested, that meant that the cause of mutation could still hide out in the herds. And the other problem is we had the potential for unaffected calves to not be identified. So as I said previously, the first symptoms of this disease aren't really shown until 8 to 14 months of age. Well, if you look at the cattle industry, if a calf is born for the dairy industry, if he's male, unless he's from a really good line, there's a good chance he's either going to be sold as a veal calf or he's going to be sold for beef. Now, if he's sold as a veal calf, he's going to be harvested before 8 months of age. If he's sold as beef, well, he's sold through a sale barn, then eventually goes to the feedlot. Currently in the United States, cattle are harvested about 14 to 15 months of age. So there's a potential that an affected calf could be slaughtered before he even shows the symptoms. And if he's in a feedlot and he begins to show the symptoms, now he's going to show that he doesn't walk as well, he's having trouble getting around. There's a good chance that the feedlot operator, looking across his thousands of animals, he's not going to say, oh, this calf has Weaver syndrome. He's going to think, the calf got hit, he's got a bruise, something's wrong, and either he'll try to give him some medication, or he'll just decide, let's go ahead and harvest this animal earlier. Even if he thinks it is a Weaver calf, because of the size and the scope of the beef industry, it's going to be really hard for that feedlot operator to tell the dairy farm, hey, I got a calf from you and has Weaver syndrome. So besides all those problems where, we have a test, but it doesn't work well, and we still have the allele hiding out in the populations, we have some different problems currently. I talked to Dave Kendall from the U.S. Brown Swiss Association, and he said, unless they don't use this microsatellite test anymore, because it was developed so long ago, and there's been so much recombination, their viewpoint is it's not effective. You have no idea if you have a false positive or false negative, and it's pretty much not useful. Also, there's been almost no research, or very little research, on this disease for the past 10, 20 years. And the reason for this is twofold. One, because of that microsatellite test, it was kind of viewed as though the problem was under control. So why do research on a disease where you already have a test? The other thing is that there were other diseases that came out. There was SMA in Brown Swiss, where it was a new, hot disease. So that's where the research money went. So historically, even though we knew that the Weaver syndrome, it wasn't solved, there was research done on it. And currently, the Brown Swiss Association views Weaver syndrome that there's a potential as a ticking time bomb. There's over 10 million head of Brown Swiss in the world, and there's a viewpoint that the Weaver syndrome, the allele frequency for it, is increasing. And so we're not really seeing an increase of affected animals currently, but there's a fear that in a couple of years, we're going to start seeing a whole lot of affected animals, and suddenly they'll be back to where they were again in the 1960s and 70s. Another problem is that Brown Swiss have been used to develop and to upgrade other breeds. So any of those breeds that has used Brown Swiss genetics, there's a chance that the Weaver allele is now in those breeds. There's actually a paper in 2008 where there was a Gyr calf in Brazil that had Weaver-like syndrome. And Dave Kendall let me know that in the 1980s and 90s, there was a lot of Brown Swiss semen shipped to Brazil. There are some Gyr farms that use the Brown Swiss. They're hoping to improve the milk traits of it. And so there's a chance that in the Gyr breed populations that there is the Weaver syndrome, and it's just lying underneath the horizon. So because of all these problems, the Brown Swiss Association asked us if we could re-look at the Weaver syndrome and try to find the causal mutation. We said we would be more than happy to. And the way we approached it is that initially we took some high-density SNP genotyping. So we used the Illumina Bovine HD chip. It has just under 800,000 SNPs. And we actually got the HD genotypes on 20 Weaver carrier animals and 51 normal Brown Swiss animals. We're calling these normal because some of them weren't genetically tested, and so we think they're normal, but we're not sure. We're actually lucky some of these genotypes came from animals that were already tested for the genomic selection, and some of them we went out and purchased the semen shards themselves into the genotyping. Once we had the genotyping, we did some quality control on it. We wanted to make sure all the SNPs had a minor allele frequency above 0.05, and we also selected on call rate. When we initially selected on a call rate of 95%, we only lost one animal. But then we went back and we looked at the GWAS results. We decided to add back one animal. So when we looked at the GWAS, the reason we used all 71 animals was that we got the highest peak on chromosome 4, which is to be expected. That's where it was before. When we looked at the call rate on chromosome 4, every animal had above a 95% call rate. Because we only had carriers and we had controls and we had normals, we did a case control with GWAS. As I mentioned, we got a really nice strong peak. We've actually been able to refine the locus from what it was historically. So historically it was a 10 megabase region with the microsatellite markers. We've now been able to narrow that down to about a 5 megabase region. And as expected, it this 5 megabase region lies within the 10 megabase area. So then we asked ourselves, well, there's been a lot of dairy cattle. Pretty much every dairy bull in the country has been genotyped on the Illumina 50K chip for genomic selection. So we wondered, well, could we actually use animals that had 50K data, do a GWAS, and get even better resolution? To answer that, I actually went in and I just stripped out the high density SNPs and I only left the SNPs that are on the 50K panel. Luckily, all the 50K SNPs are represented on the HC chips, and so I went back and I looked at just those 71 animals again and found out if you only use 50K SNPs, you actually get the same resolution as with the microsatellite markers. So you're back to about a 10 megabase region. The other problem is that your peak is actually moved to the side. So the 8 high density SNPs, we actually have a peak that's kind of on the left side of the 10 megabase region. If you use all the 50K SNPs, what it looks like is that your peak is more to the right. We also though wondered, after looking at SNPs and the GWAS, we're wondering if this region of chromosome 4 is misassembled. Pull out LD on about 20 breeds, and then I just pulled up and I said, okay, I want to look at just this region on chromosome 4. So if we had had a misassembly, our thought is that we would have LD, so bright reds, all the way down here on all the breeds. If there was a misassembly, if it was a region that was flipped, the LD should show that there's 2 regions that are far apart than in high LD, but the regions in between are not in LD. We really did not see that on any of the breeds, so we have reasonable confidence that this region of chromosome 4 has been assembled correctly. So now that we're confident that everything is assembled, we know the area to look at, now we decide to move into trying to identify the causal variation. So we want to know, what mutation causes a normal brown Swiss cattle to be a Weaver calf. And for that, we have to do sequencing. And so the question is, how are we going to do sequencing? We're lucky today to have a lot of options. There's whole genome sequencing, there's exome sequencing, there's targeted sequencing. And so we actually decided to use whole genome sequencing. At the time of this study, we had just gotten an Illumina high-seq machine, but the indexing had not been commercially released yet. So we decided to use a pooling technique. So we took 10 normal animals and 10 Weaver carriers. All the carrier animals were actually carrier identified from progeny testing. So we didn't have to worry about, are these false positive animals. We made 300 base pair insertion libraries, and we sequenced them on, we did a 2x100 paired-in sequencing on the Illumina high-seq 2000. And then we had a couple lanes open up on our Illumina GA2X. So the Weaver carrier animals also had read 120 base pair reads on that. Steve Stronger, one of our bioinformatics, took all that sequence data and he mapped it to the University of Maryland's 3.1 assembly. He used BWA to map it, and then he used what's called GATX for local realignment. The GATX is a program from the Broad Institute, and what it helps with is to have proper local alignments with indels, with insertions and deletions. After he mapped it, we had about 25 to 30x coverage of the entire genome, and then we looked within that 5 megabase region. Within that 5 megabase region, we had over 53,000 sequence variations. Most of these were single nucleotide variations. There are some insertions and some deletions. So now we have 53,000 variations. We need to find the one mutation that actually causes Weaver. So for that, we had to filter. Since we had a pool, the way I filtered these was I said, okay, I need to have that minor allele in the normal pool. It needs to be at or above 90%. And the alternative allele I can see once. The reason I didn't say the minor allele frequency had to be zero, I couldn't see any of the alternative allele, is I wanted to allow for any sequencing errors or potential misassembly on one or two reads. Basically, I didn't want to be too conservative and potentially toss out the cause of mutation. For the Weaver pool, since this was the pool, we said the minor allele frequency had to be at or below 70%. Just in case with the sequencing, we, instead of capturing the cause of mutation, we were capturing the historic brown Swiss allele most of the time. So we allowed for that minor allele frequency to be a little bit higher than most people would have done. And then we said, well, Weaver syndrome is only in brown Swiss cattle. So if we see an allele and it's present in other cattle breeds, that can't be the cause of mutation. And of course, part of the logic behind this is that all the different cattle breeds, they diverged a long time ago. The Weaver mutation in the U.S., we assume it came about in the early 1900s. All the cattle breeds today, they've been diverged for 200 to 1,000 years. So we actually took animals that had been sequenced for the SNP discovery projects that our group had in Mark. We took sequence from some dairy breeds, some beef breeds, and some Bos indicus breeds. And we said, if I have an allele from the sequencing and it's found in the sequence from these other breeds, toss it out. And then finally, we filtered against repeat regions. From all of the studies, the cause of mutation is only located on chromosome 4. If there's a repeat region that's spread across the genome, that's not going to be it. Plus, if it's a repeat region, sad to say, it's going to be really hard to develop a diagnostic test for it. After all this filtering, we identified 116 Weaver syndrome or Weaver carrier unique sequence variations. So we went from 53,000 variations down to 116. After that, we passed it through a program called SNP-F to estimate the effect of those variations on genes. Through SNP-F, of the 116 sequence variations, it estimated that based upon the current University of Maryland 3.1 annotation available at that time, that there 71 of those SNPs were energetic, 16 were flanking, so potentially in the untranslated regions of the gene, and 3 were microsatellite. Within a gene, 22 of them were intronic, 1 was a synonymous amino acid change, and 3 of them were predicted to have a non-synonymous change. Sorry, non-synonymous amino acid change. So from this, we decided to hedge our bets, and we said, you know what, most likely, given the history of the Weaver syndrome, the mutation that's most likely to cause it is going to be a mutation that's within or close to a gene. So we took all those ones within a gene, and through a collaboration with GeneSeq, we developed a multiplex SNP genotyping. Sadly, one of the intronic SNPs, the reaction failed for it. But we developed this multiplex assay, and we decided to test brown Swiss animals. Throughout the past couple of months, we have been collecting brown Swiss samples from across the U.S. and from Europe through some of our collaborations. So we had over 800 brown Swiss animals to test. We had been lucky to identify 2 affected animals, a whole bunch more carriers, and a whole bunch of normals. We also wanted to make sure that this mutation is not in other animals and other breeds. So we identified some non-brown Swiss animals that we DNA. We decided to test them as well. So after we did all the genotyping, we re-imported those genotypes into Golden Helix. We updated our marker map, and we did a combined GWAS study. So the combined GWAS is a middle graph, and we got some great results. We're up to a negative log 10p of above 70. So this is nice. It looks like, you know, we're definitely looking at the correct region. We initially saw this, it's like, yes, you know, we potentially have the cause of mutation. But then we actually went back and looked at the actual genotypes. So I know it's a little busy, but we actually pulled out is that there are 3 SNPs that look promising. So we're looking at the 2 animals that are Weaver affected, WW, animals that are Weaver carriers from progeny testing, animals that are identified as Weaver carriers from the genetic test, non-carriers from genetic testing, and then a non-carrier just because they hadn't been tested. Everything looked great. These 3 SNPs, they're homozygous for one allele in the affecteds. They're pretty much heterozygous in all the carriers, and they're pretty much homozygous for the other allele in the non-carriers. But there's a slight problem. One of our carrier animals is homozygous, and 3 of our non-carriers or 3 of our normals are homozygous. So why is this? Why is it we have 4 animals that are not supposed to be affected animals, but they're homozygous? Well, we looked at, there could be a couple explanations. One of them, there's just a genotyping error. So we went back and we re-genotyped those animals, and then we we looked at the signal strength of those SNPs. Turns we got the re-genotypes back. They genotype the same way. They're homozygous for that A allele. And looking at the signal strength, everything looked good, so we believe these are true genotypes. So another possibility was that these animals are misidentified. Either these are animals that were, you know, the wrong DNA got put into the wrong well, or it's just these are animals that actually were affected, but something happened and they were harvested or they died before the syndromes showed themselves. Or it could have been that they had the disease, but it wasn't reported. This is a possibility, and we're looking into it. The other possibility is, you know, we had 116 variations, and we only tested those that were predicted to be in or close. There's a good possibility that some of these energetic genes could be the cause of mutation. It could be that there's actually a microRNA there, or it could be that there's actually a another exon of a gene that's farther downstream. It could be that there is a small open reading frame. There's a paper that just came out in fruit flies where they improved the algorithm for predicting genes and for identifying the small open reading frames. And in fruit flies, they identified there's 400 to 1,000 new small open reading frames. So these are genes that encode proteins that are less than 100 amino acids. There's a really good chance that one of these predicted energetic genes is actually a mutation within a small gene that just hasn't been annotated yet. So we actually are going, we currently are developing multiplex assays for those energetic SNPs and the microsatellite SNPs. And we're going to retest over all those animals, and hopefully one of those will be the causal mutation. You know, if it's 100% concordant with the phenotypes, that's great. If it's not, then we will see if we can develop a diagnostic haplotype. And that haplotype can then be used as a genetic test for the farmers. So the benefit of if we either identify the causal mutation or diagnostic haplotype is we will be able to present to producers a diagnostic test that they can use. If we identify the causal mutation, this can be used in all future generations, and it can be used in other breeds. And because of this, that potential ticking time bomb will be removed. Sorry about that. The other benefit is that this will allow producers to manage their herds better. They won't have to toss the baby out with the bathwater. If they have an animal that is a Weaver carrier, they actually can then go out and test all their females. They can identify females within their herd that are normal, that are non-carriers, and use that bull to mate with them. Yes, this will still produce about 50%, or this will produce 50% carriers as an offspring, but if that bull is a really good producer for milk fat, for reproduction, that way you don't have to toss out all those good genetics just because of one bad gene. Because of this, you'll be able to manage it properly. With that, that's the end of my talk. I'd like to thank everyone that has helped with this project, both within the USDA, also with the Brown Swiss associations, our European counterparts that have helped us get some of the Italian DNAs. Really like to thank GeneSeq for their help with the sequenome testing. Also to thank Frank Nicholas from the University of Sydney for some advice, and really like to thank Go and Helix. Throughout this presentation, what I kind of really didn't hit on was how quickly we were able to do the analysis. I was able to do a GWAS in 5 minutes once I had all the data in there. Once I had the other targeted genotypes, I was able to quickly put all the genotypes into the program and rerun the analysis. It's really been helpful having these advanced softwares to help analyze the data quickly. And with that, I'll take any questions.
All right. Well, thanks, Matt, again for the presentation. As I said at the beginning, there is a question and answer pane on the GoToMeeting window. It should be on the right side of your screen, unless you moved it. Go ahead and enter your questions there. We have been getting quite a few questions throughout the presentation, so I'll start with those. But it looks like we got another 15 minutes here, so we can answer plenty of questions in time.
Okay.
First question for you, Matt. The person asked, breeders experienced in the last 50 years that selection based on theory of population genetics is very effective. Do you think that finding several causal mutations will increase genetic profit or profit rate in breeding programs?
If it's causal mutations for diseases or causal mutations for phenotypes. If it's causal mutation for diseases, it'll help their profit somewhat, but that's mainly because they won't have these horrible diseases. Case in point, in the Angus Association, there's a curly cap disease. John Beaver, within a year, identified the causal mutation, and they are now able to properly manage that. The benefit of the producers is they don't have calves that are born dead, so that increases their profits. As far as phenotypes, as far as milk yield or the size of your steak, to quote my boss Kurt Van Tassel, we don't need no stinking genes. We are actually able to make genomic selection without using the causal mutations. We're able to use the SNPs and build prediction models that tell us how well an animal will perform, either for production phenotype or even in the future for disease resistance. In that case, this genomic selection that will improve a producer's paycheck, either because the animal will be able to have better milk yields, or because the animal will be selected to have better feed efficiencies. But the way we look at it is, we're providing tools and data for the producers, so we'll tell them how an animal will perform for certain phenotypes. Then they are able to use that data to select what is best for their region and their management.
Okay. A follow-on question to that is most of the SNP effects on quantitative traits, for example, milk yield, are extremely small. Is there any reliable statistical method proving that certain SNP is significant influence in the genetic variation of the trait?
I'm not sure how to answer that. I would say yes, it's been proved. These have been used for the past 3 years in the dairy industry, the SNP effects. If you look at any quantitative trait from height to milk yield, my viewpoint is that there are hundreds of variations that affect it. If you look at growth, pretty much any gene in your body is going to affect growth. If you can't eat as well, if you can't digest your food as well, you won't grow as well. So what we have is we have a SNP that has an association with the trait. So we can show that yes, it's there. There are studies being done currently, I know in the beef cattle and I believe in the dairy industry, showing that the improvements of genomic selection, showing that a herd that is not used genomic selection and a group that has had genomic selection done on it. There are multi-year studies going on right now showing, hopefully at the end, showing that yes, the genomic selection really does work. My viewpoint is good that we have these, but I think they're going to be real. These SNPs are going to be associated with some causal mutation or a causal region. We don't actually need to have the causal mutation for phenotype, for milk yield. We can use SNPs that are associated with it that are in high LD. So, yes, they work. There will be data that come out that shows it work, but for quantitative traits, I see no reason not to trust it.
Okay, here's a rather simple one. When you show the diagrams of SNP effects, what values are presented on the Y scale? I believe that's with the GWAS plots.
Oh, that is negative log 10p. For the SNP effects, if you're looking at the GWAS, so I apologize for that. On the y-axis, this is negative log 10p.
Okay. Question here is what about derived and ancestral alleles? That's the extent of the question, but
can you identify them? Yes. For the phylogenetics studies, Jared Decker, he was able to identify the ancestral allele. That's one that he was able to use the phylogenetics. I know that there are people using these SNP chips to identify admixture, to identify Bos-Taurus integration into other species. And for that, they identify the ancestral allele. So, if you take two breeds, if you take a water buffalo and Bos-Taurus, well, you can identify, which allele is the common one, which is the ancestral allele. And from that, you can identify which allele is the non-ancestral allele. And the benefit from that, then, if you have any population you think that has admixture, let's say it has a low level of Bos-Taurus integration, and you want to remove that, you actually then can identify and select animals that have the lowest level of integration.
Okay. Moving on here, for the GWAS that you did, how related to each other were your samples? And did you do any correction for possible population stratification? Is this capability built into the software?
For the GWAS, pretty much what it was at that time was that we had 71 animals with genotypes. So, yeah, they were related. Any dairy animal is going to be related. When I did the sequencing, I selected the animals that were as unrelated as possible. I did that to increase the amount of genetic variation. But for the GWAS, we had 71 animals, and I used them all. For the stratification, I think, Josh, you can answer this better, but if I recall correctly, yes, it can. Genomes, Golden Helix can take into effect stratification.
Yes, so there are a number of quality assurance and other statistical analyses beyond just the association tested in the software. Population stratification is one of those, and we have a number of researchers who are doing not just human genetics but plant animal genetics that do use the population stratification within the software to correct for that. Another question here, would you stick with the HD chip if you were to do the same project over again, or would you do more animals on the 50K chip to potentially capture more historical recombination events?
For a causative disease mutation, so I actually had, or for a disease, so you had a talk with John Bieber a couple months ago, kind of addressing this. So he's had a lot of success using the 50K chip. And from my talks with him, what I believe is that if you have a newer disease to where it's, the cause mutation appeared relatively recently, I think you could get some good results with the 50K chip. If you're dealing with older mutation, and from what this is shown with the, this is at least 100 years old or not older, I think the high density chip gives you better resolution. The 50K chip, as I showed, it's the slides up here, it'll work. It does show you the right region. It gets you close. Probably that's not the right one, but the problem is then you're looking at a much bigger region. So, for here, with the 50K chip results, I'd be looking at a 10 megabase region. I could potentially even say, well, I think it's probably over to the side. With the HD, at least I can narrow that down. So, yes, you can use the 50K. The HD is just going to give you better resolution. So that just comes down to your time and money and your studies to what you think.
Okay, I've got a couple questions. Oh, I'm sorry, go ahead.
Oh, go ahead.
I just have a couple questions here on copy number analysis. One person asks, have you looked for copy number or other structural variants from both HD and sequencing data? Another person asks, human studies are examining the role of copy number variation in ALS or Lou Gehrig's disease. What are your comments on looking for copy number variations in these?
So we did look at that a couple months ago. So one of the scientists in our group, George Lee, his group is one of the leaders of copy number variation in cattle. And we actually asked them, hey, do you see any copy number variations in this region? And they did a quick look at the data they had and said, nope. So from what they saw, there was not a CNV in this region of brown Swiss. They were looking at both 50K data and more from the sequencing stuff. So they really didn't see anything in that. It was a couple months ago, so they may have more. But from what we're seeing, or from what we've seen so far, it's not CNV. Now, if these next SNPs that we test, if none of them are 100% concordant, yes, that's something we'll come back and look at. Unfortunately, because we did pooled sequencing, we were kind of eager to do the sequencing, and we didn't wait a couple of months until the indexing came out. Because of that, we really can't look at the CNV as much because our sequences are not individual animals; it's a pool. But yeah, if we don't get anything, then CNV analysis would be something else to look at later on down the road. So
okay,
hope that answers it.
Yep. Another question, I guess I can answer this one, is can the Golden Helix software be used for other species beyond humans and cattle? The answer to that one is yes. We do have a number of, we can really support almost any genome. Software is really designed for diploid organizations, but we do support a number of plant animal as well as the human species, and you can do that all within the software. If there is a particular genome that we can't support, there are ways automatically with the software, there are ways to import the genome and curate the annotations as well if you're not studying cattle or humans. Or some of the other genomes that we do provide. I believe in the USDA there are a number of people studying other species beyond cattle. I'm not sure, Matt, has anybody in your group used it for other species beyond cattle at this point?
We actually have a visiting Brazilian scientist who's doing sheep research, and so she's actually using it for the sheep. was 2 months ago she started. At that time, Golden Helix did not have the sheep genome up on their website. I sent them the email, I think in 2 days. They had the Illumina SNP chip for sheep, that map up along with the annotation. And we do have people, I know we have collaborators looking at goats, so once the goat stuff comes out. Yeah, pretty much my experience has been if your annotation is available and it's not up, just ask Golden Helix. If you've bought the software and using it, in a couple of days, they will have it up and going.
Okay. We've got a couple of minutes here left for the presentation. I've got many more questions. We're not going to be able to get to them all. I'll maybe answer or ask one more. But we do have these questions on file, so we'll make sure that we either send them to Matt to get them answered, or we can answer some of them ourselves. I guess the last question here, Matt, for you is have you published these results yet? Somebody asks.
No, we have not. We have not, and we hope to. As some of you may have noticed, there were no gene IDs up there. We do know there are other labs in the world that are so interested in this. So we intentionally did not put up some of the gene names. We hope to publish this as soon as possible. As soon as we get back to those next genotype results, we'll kind of decide from there. We'll look at it, and if we think we have a good enough story, we'll publish as soon as possible.
Actually, so a follow-on question to that is how would you leverage the commercial value from the work that you are doing? Would you ordinarily patent your SNPs? Would you wait to identify the causal mutations, or would you use these other methods, not including patenting?
So what's been done with other people, I know there are other groups that they have found causal mutations, and they have kept them as company secrets. They'll have genetic tests. Some people patented it. Our viewpoint of the USDA is that we want our research to have the largest impact as quick as possible. So if I recall correctly, right now we're not looking at patenting it. We just want to get the test out. We've had a lot of help from Brown Swiss Association, and we'd like to pay that back to them. As far as for patenting causal mutations, commercially there's a need for it. It does help the research, but my viewpoint that if there's a way to really help their producers, whether they patent or not, get that data out there for them, let them use it. So yeah, that's kind of my answer.
Okay. Well, so we've run up to the end of the hour here, and I want to thank you again, Matt, for the presentation today. Again, there are a number of questions that we couldn't get to, and we'll try to do those by email. Of course, if you have any questions that you would like answered, I believe, Matt, people can call you up if they wish, and we're certainly willing here at Golden Helix to answer any questions that you have on the software, or perhaps what other people are doing research-wise with our software. We'd be happy to answer those questions as well. So again, thanks, Matt. Thanks, everybody, for coming, and hope it was helpful for you. With that, I'll go ahead and sign off.
All right. Thank you much.